Hello, in this video we will discuss about the fatty acid synthesis in prokaryotes, for example bacteria. So in this way, basically we will also learn about the phospholipid synthesis. So let's begin to understand. First of all, we are understanding the bacteria prokaryotes, which that is bacillus, bacilli bacteria, the bacillus subtilis, which that is basically important, the phospholipid synthesis as well as the fatty acid synthesis, because the first is the fatty acid synthesis is very important for the production of long chain of fatty acid, while the phospholipid is the phosphate group attached to form hydrophilic and hydrophobic interaction in the bilayer of phospholipid of the plasma membrane of the bacteria. So the bacteria plasma membrane synthesis due to phosphate group addition with fatty acid synthesis cycle and by phospholipid synthesis. So let's begin to understand without wasting any time. We are going to the fatty acid synthesis first, and after we will understand about the phospholipid synthesis. So let's begin to understand. For example, the glucose containing medium are present in the bacteria. So the bacteria allow to move influx of the glucose to by a different mechanism. We are not going to understand with complex information. So the glucose will enter the GLUT channel through GLUT channel. In this way, the glucose will enter into the cytoplasm, and here is the without oxygen the glycolysis will be occur, because the bacteria not contain mitochondria, so the aerobic respiration will not occur. So this glucose in the under the glycolysis to produce ATP and 2 NADPH to form a 2 molecule of pyruvate from the one glucose molecule, because the each pyruvate contain 3 carbons. So in this way, the ATP and hydrogen bicarbonate will be used to as well as the acetyl CoA carboxylase A, B, C, and D types, which that can be used to produce malonyl CoA, can be inhibit the FAP or. So this when release the ACPSH, basically when add in this way, you can understand the ACP will be bound while the CoASH will be released. So remember, the CoASH will be add, so the ACPSH will be released. So the reversible. So in this way, this is the fatty acid. Fatty acid is here is addition of the acetyl CoA, and the of water will be released. The fatty acid synthesis to form this fatty acyl CoA formation, and this fatty acyl CoA will be added into the fatty acid synthesis cycle. So in this way, here is the basically is the 1 2 3 carbon containing molecule contain ACP will be add with the fatty acyl CoA to CoASH release and carbon dioxide, and the FabHA and FabHB in the third A step. In this way, it will be form this structural formula, which that contain R, the alkyl group, and here is the CH2 containing 1 carbon, and the 2 carbon is this type, and the condensing enzyme transacylase formation will be occur, and in this way the 3B will be FabF is used to form a ACPSH, and in this way the release the ACPSH plus carbon dioxide. And in this way, here you can see this is the ACP will be formation in the carbon number 4, and in this way the 2 carbon further increase will lead to 4 carbon, and in this way the FabG will be fourth step will lead to NADPH will be form, and in this way here you can see this is the formation of the NADPH. Hydrogenation will lead to increase the hydrogen, so in the oxygen level, and in this way you can understand the water will be released in the FabZ enzyme in the fifth step to form. Here is still ACP are present, and in this way you can see the double bond will be occur. After this, the double bond between the carbon to carbon double bond, it will be unsaturated fatty acid formation, but in this case here is the NADPH will be used to produce in the presence of Fab1 condensing enzyme transacylase. So in and Fab L will produce this type, and in this way you can understand the again this formation, and in this way the addition of the carbon is the basic mechanism. This is we are just understanding about the addition of the carbon in the fatty acid, and in this way the addition by this mechanism the phospholipid synthesis will be occur, and in this way the increase the carbon number due to the fatty acid cycle. So after this the phospholipid synthesis we will discuss now. So in this way here is the after the addition of the carbon, here is the phosphate, and ACP will be released through a phospholipase X7. So in this way here is the containing basically the phosphate group will be added where the ACP will be was added. So in this way you can see the glycerol 3 phosphate will be used to separate the phosphate group, while the glycerol will be added through a phospholipase gamma in the 8th step. To form this glycerol will bind with the two fatty acid where the phosphate group are present basically, but it is not to one only one chain of fatty acid, but it will be along further acyl ACP add more to add the phospholipase C to produce two second chain of the fatty acid. In this way the two chain will be combined together with the carbon number 2 and carbon number 3 of the glycerol. In this way the two chain will be formed, and in this way you can understand this is the hydrophobic and hydrophilic interaction will occur through a phospholipids bilayer. Inside is the fatty acid chain which that is the hydrophobic, while the phosphate group is basically used for the hydrophilic. So this interaction is due to the this formation because phosphate group basically attract the water, while the inside is the double chain, the unsaturated and saturated fatty acid formation will lead to hydrophobic interaction. So anyhow, further is the pyruvate also used for the fermentation like ethanol, alcoholic fermentation, the acetobacter used for the acetic acid fermentation, methanogen is used after the releasing of the carbon dioxide, the methane gas production. So this is the different mechanism, also the anaerobic fermentation, not aerobic fermentation. So that was the overview about the fatty acid cycle, synthesis cycle, and the increasing of the carbon. Increasing of the carbon means the carbon when increased to increase the chain of fatty acid is used for the phospholipid bilayer. To increase the chain will lead to more hydrophobic interaction inside of the plasma membrane for the semi-permeability and the inhibition of that molecule which that is the hydrophilic, while the hydrophobic basically can be directly moved like steroid or other things. So this is the phospholipid synthesis as well as the fatty acid synthesis cycle. In this way the phosphate group is add will lead to basically after the ACP second ACP will acyl second ACP will be add. So in this way you can understand. Thanks for watching. Please make sure to subscribe, like and share. Bye.